This is a podcast of the Nuffield Department of Medicine. We spoke to Dr. Christopher Yao about his research on big data. Hello, Chris.
Hi.
Can you explain what big data is?
Big data is a phenomenon which has really arisen out of a long period of data-driven science. In genetics, what we've seen is that the cost of genomic technologies has rapidly reduced in cost over the last decade. And at the same time, the number of samples and people that we're analyzing has rapidly increased. And in big data, what we're trying to do is look at the massive data repositories that we've generated over the last 10 years and will be generating in years to come, and trying to extract knowledge from these data sets to not only answer just one particular scientific question, but possibly multiple scientific questions as well.
How is big data helping you explain differences in cancer?
Perhaps I could start with an example. My group works with the Ovarian Cancer Laboratory in Oxford. And in one of our studies, what we've been able to do is to take 40 tumour samples from a single ovarian cancer patient. These tumour samples have come from different parts of the body where the disease has spread. And we've taken these samples at different times during the patient's diagnosis. We've also taken samples before and after chemotherapy. And what we've done is to sequence each of these 40 tumour samples, and in the process generate a massive 40 terabytes of raw data for this individual patient. And what we've been able to learn from this data is critical insights into how the cancer arose in this particular individual, how it evolved and spread in that individual, and how this tumour reacted to the chemotherapy. And this has been really important because this comprehensive profiling has allowed us to gain insights into the tumour evolution in this patient that we couldn't have got from just a single tumour sample. And what's going to be really exciting in the next few years is that we're going to be applying the same technique to further ovarian cancer patients and putting together a very massive, comprehensive profile of ovarian cancers in different patients.
Why is it important to understand these differences in tumours?
Patients respond differently to cancer treatments, and at the moment we don't have a full understanding or the ability to predict exactly how they will respond. By looking at the genetic differences between the cancers and genetic differences between cells within the same tumour as well, what we're trying to do is learn about the mechanisms of drug resistance and also of radiotherapy resistance in these patients. So by relating the genetic changes to how they respond, what we're hoping to do in the future is be able to develop better and more effective treatment plans for these patients.
What are the most important lines of research that have risen in the past 5 or 10 years?
What's been really exciting in the last 10 years has been the convergence of two different fields. On the one hand, we have statistics, which is traditionally a mathematical discipline, and on the other hand, we have computer science, which is more technologically driven. And as these two fields converged in the field known as machine learning, what we've seen is an explosion of new ideas for analysing data and developing smarter, more efficient computational algorithms. And this has been really important because in parallel, in genetics, we've seen an explosion in the amount of data we can collect and generate. And without these new ideas coming from machine learning for interpreting this data, we would have the means of generating lots and lots of data, but no means of understanding it.
Why does your research matter, and why should we put money into it?
One of the most interesting things which has occurred in this year has been the announcement of the Genomics England 100,000 Genomes project. Now this project is going to be very important because it will sequence 100,000 genomes and provide another resource of data for us to study. But one additional implication is that it will also help the NHS prepare for the challenge of integrating genomic technologies into modern healthcare. However, whilst it's easy to sort of buy more sequences to meet the sort of capacity challenge of integrating genome sequencing into healthcare, it isn't quite so easy to hire and train new data analysts. So a lot of my work is concerned with developing machine learning algorithms that make the task of processing and analysing complex genomic data sets much easier and much faster. But it's important also to consider that machine learning in a biomedical context is a lot different to machine learning in other applications. So most people are probably familiar with the use of sort of automatic face tagging in social networking sites or speech recognition software on their phones. But in these applications, if you make an error, it's generally not a terrible thing. In fact, it might be quite amusing. But in a clinical context, we can't afford to make errors. And we need to engineer machine learning algorithms that respect much more stringent specifications for robustness and reliability.
How does your research fit into translational medicine within the department?
Ultimately, what we would like to do is to turn the machine learning algorithms that we develop and make them from research tools into clinical decision-making support systems. For example, we've been working with the Biomedical Research Centre here in Oxford to develop a diagnostic system for leukemia, which allows us to translate complex genomic data coming from a particular type of technology into a simple 1-2 page report that can be used by clinicians to develop a patient treatment plan. And in the future, this automation is going to become more important because we will see multiple genomic technologies being used, and also the need to integrate these genomic technologies with imaging technologies and other types of information that are being gathered about patients.
Thank you, Chris.
Thank you.